Huge sequoias rise out of the earth, often climbing hundreds of feet into the sky and weighing hundreds of tons. What is the process that creates these huge biological monoliths? Where does the matter that creates their mass come from? The earth from which they rise? The air that surrounds them? The rain that falls on the forest? And what provides the energy to build them, year by year, molecule by molecule? Experiments by the 17th century Dutch physician Jan van Helmont indicated that the soil in which a tree is grown contributes little to its total mass. Van Helmont planted a seedling in a container, carefully weighing the soil in the container before planting the seedling. He then proceeded to care for the seedling for 5 years. After the 5 years, he weighed both the seedling, which by now had become a small tree, and the soil in the container. During the 5 years, the mass of the tree had increased 75 kilograms, while the weight of the soil was nearly unchanged. The only substance that Van Helmont had added to the container had been water, so he hypothesized that it is water that provides for the increase in mass that occurs in trees and other plants as they grow. As we will see shortly, Van Helmont was only partly correct. Nearly 100 years later, scientist John Priestley performed an experiment that indicated that plants give off a gas essential for combustion, oxygen. Later, the Dutch scientist Jan Ingenhousz noted that plants only give off oxygen if exposed to light. Even later, scientists discovered that trees and all other forms of plant life, in fact, all forms of life, were made up largely of molecules that contained carbon atoms. Where did all these carbon atoms come from? Most soil has little or no carbon, and water certainly has none. The only likely source was the carbon in the gas carbon dioxide, which is one of the gases that makes up the Earth's atmosphere. As a result of the experiments by Van Helmont, Priestley, and Ingenhousz, and more recent scientists, it became obvious trees and other plants obtain the matter and energy needed to sustain growth through chemical reactions involving water from the soil and carbon dioxide from the atmosphere, and that these reactions are powered by light energy and produce oxygen as a byproduct. In addition, the process produces molecules made out of the carbon, hydrogen, and oxygen atoms provided by the water and carbon dioxide molecules. These are the molecules that provide the energy and matter needed to fuel plant growth. Scientists discovered that all of these molecules have the general chemical formula CH2O multiplied by N, where N is the number of carbon atoms. As a result, scientists named them carbohydrates, which literally means carbon plus water. One of these carbohydrates, glucose, has the chemical formula C6H12O6 and is one of the molecules most often formed by the process or series of chemical reactions scientists now call photosynthesis. Since it takes 6 CO2 and 6 H2O molecules to make a single glucose molecule, the equation for photosynthesis is usually written as 6 CO2 molecules plus 6 H2O molecules plus light energy yields 1 glucose molecule, C6H12O6, plus 6 O2 molecules. Nearly all life on Earth is supported, directly or indirectly, by photosynthesis. Glucose molecules formed during photosynthesis are broken apart in the cells of plants and animals in a series of chemical reactions called cellular respiration. Cellular respiration reverses photosynthesis. This is reflected in the chemical equation for cellular respiration. 1 glucose molecule plus 6 O2 oxygen molecules yields 6 carbon dioxide molecules, 6 water molecules, and a release of energy. The chemical energy stored in glucose molecules can be released via cellular respiration, either by the producing plant itself or by animals consuming the plant or its fruits and nectars. Glucose not consumed for energy by the plant producing it or by animals is often joined in long molecular chains to form the material cellulose, which is the major structural component in the cell walls of plants and thus an integral part of plant structures such as tree trunks and branches. Let's now take a closer look at how the process of photosynthesis is carried out by plants. As alluded to earlier, photosynthesis is a process or series of reactions which use the energy of sunlight to convert the low-energy reactants, water and carbon dioxide, into high-energy glucose molecules and oxygen. In plants, photosynthesis takes place in cellular organelles called chloroplasts. In most land plants, the majority of chloroplasts are located in the cells making up a plant's leaves, where they can easily obtain the light energy necessary to power photosynthesis. The leaves of most plants are only a few layers of cells thick, ensuring that light energy reaches even the leaves' innermost cells. The upper and lower surfaces of leaves are covered with a layer of transparent cells called the epidermis. The epidermis cells are transparent in order to allow light energy to reach the cells in the middle layers of the leaf where photosynthesis takes place. A transparent, waterproof, waxy layer called the cuticle covers the epidermis. This prevents one of the reactants critical to photosynthesis, water, from evaporating from the leaf when weather conditions are hot and dry. Adjustable pores, called stomata, control the flow of the second major reactant, carbon dioxide, into the leaf and the flow of water and one of the products of photosynthesis, oxygen, out of the leaf. Between the epidermis and the cuticle that cover either side of the leaf lies the mesophyll, or middle of the leaf. The mesophyll is made up of a few layers of cells that contain the vast majority of a leaf's chloroplasts. Vascular bundles, or veins, supply water and minerals to the cells that make up the mesophyll and carry sugars that are produced in their chloroplasts to other parts of the plant. The leaf cells of plants usually contain a substantial number of chloroplasts in their cytoplasm. Chloroplasts are isolated from the cytoplasm by two membranes. Inside the membranes is a semi-fluid medium called the stroma. Contained within the stroma are disc-shaped, interconnected membranous sacs called thylakoids. The thylakoids in most chloroplasts are usually placed one on top of the other in stacks called grana. The simple chemical equation for photosynthesis that we saw earlier hides the fact that dozens of chemical reactions controlled by large molecules called enzymes are necessary to complete the process of photosynthesis. But at its essence, photosynthesis can be thought of as being divided into two different groups of reactions: light reactions that depend on light energy to power them, and dark reactions, which are powered by molecules formed during the light reactions. The process of photosynthesis begins with the light reactions which occur in the membranes of the thylakoid sacs, which are also often referred to as the photosynthetic membranes. Chlorophyll and other pigment molecules in the photosynthetic membranes capture light energy and convert part of it into chemical energy, which is stored in energy carrier molecules for later use in providing the energy for the dark reactions that occur in the stroma. Before going further, let's take a closer look at the nature of sunlight, which usually powers the light reactions. As you probably know, the sun emits a wide spectrum of electromagnetic radiation. At one end of the electromagnetic spectrum are very high-energy gamma rays, X-rays, and ultraviolet waves. On the other end are longer wavelength, lower energy, infrared, micro, and radio waves. Visible light, lies between these two ends of the electromagnetic spectrum and contains the wavelengths of electromagnetic radiation most useful to living organisms. What looks like white light reaching us from the sun is actually made up of many different colors, corresponding to many different wavelengths. Visible light waves extend from the ultraviolet and blue range on one end into green, yellow, and red up to the edge of the infrared range on the other. Light striking a leaf can be reflected, transmitted through the leaf, or absorbed by it. The light reflected or transmitted by a leaf produces its color, while light that is absorbed heats up the leaf or drives biological processes like photosynthesis. In biochemical terms, pigments are any natural substance that occur in and color the tissues of an organism. Since chlorophyll, the key light-capturing molecule in photosynthetic membranes, is largely responsible for the green color of most plants, it is often referred to as a pigment. Other molecules, such as carotenoids and phycocyanins, are also often present in photosynthetic membranes, capturing light energy and transferring it to chlorophyll molecules. Carotenoids and phycocyanins are referred to as accessory pigments. Each pigment, or accessory pigment, absorbs and reflects different wavelengths of light. Chlorophyll absorbs violet, blue, and red light, but reflects green light, thereby giving leaves their green color. Carotenoids absorb blue and green light and reflect yellow and red, while phycocyanins absorb green and yellow and reflect blue or purple. During fall, it is the color of the carotenoids and phycocyanins made visible by the disappearance of chlorophyll that gives leaves their dramatic yellow, orange, red, and purple colors. As all wavelengths of light are absorbed to some degree by either chlorophyll, carotenoids, or phycocyanins, all wavelengths of light can drive photosynthesis to some extent. But the peak wavelengths lie in the blue and red regions of the visible light spectrum. Let's now look at the cellular structures, photosystems 1 and 2, that begin the process of converting light captured by pigments into chemical energy. Within photosynthetic membranes, chlorophyll, accessory pigments, and electron carrier molecules form highly organized units called photosystems. The photosynthetic membrane of each thylakoid contains thousands of copies each of two different kinds of photosystems, photosystem 1 and photosystem 2. Both photosystems consist of two major parts, a light harvesting complex and an electron transport system. The light harvesting complex of either photosystem contains roughly 300 chlorophyll and accessory pigment molecules. These molecules absorb light and pass the energy to a specific chlorophyll molecule called the reaction center. The reaction center is located next to the electron transport system, which is a series of electron carriers embedded in the photosynthetic membrane. When the reaction center's chlorophyll molecule receives enough energy from the other molecules in the light harvesting complex, one of its electrons absorbs the energy and is ejected from the reaction center's chlorophyll molecule and over to the electron transport system. This energetic electron is passed from one carrier to another along the transport system. At some of the stops, the electron releases energy that is used to drive the synthesis of energy-carrying ATP or NADPH molecules. A photosystem 2 is always linked in series to a photosystem 1. The major difference between photosystem 1 and photosystem 2 is that photosystem 2 generates ATP molecules, while photosystem 1 generates NADPH molecules. Let's take a look at how the two photosystems are linked and how energy-carrying ATP and NADPH molecules are formed. As we saw earlier, light energy is captured in photosystem 2's light harvesting complex, and the energy carried to the reaction center. After the reaction center receives enough energy from the light harvesting complex, an energized electron is ejected from the reaction center to the electron transport system of photosystem 2. As the electron passes from molecule to molecule, it releases energy that is used to pump positively charged hydrogen ions across the photosynthetic membrane. As a result, the thycaloid compartment becomes positively charged, while the surrounding stroma outside the membrane becomes negatively charged because of the loss of the positively charged hydrogen ions. This creates a store of potential energy that will be used later as positively charged hydrogen ions rush back out of the thycaloid compartment through channel proteins due to their electrical attraction to the negatively charged stroma outside the membrane. Enzymes attached to the channel proteins use the energy created by the flow of hydrogen ions to create high-energy ATP molecules by adding a phosphate-based molecule to ADP molecules. Meanwhile, light rays are also striking the light harvesting complex of photosystem 1, and the energy captured is causing an energized electron to jump from the photosystem 1 reaction center to the photosystem 1 electron transport system. The photosystem 1 reaction center immediately obtains a replacement for its lost electron in the form of the last electron down the photosystem 2 electron carrier. Photosystem 1's high-energy electrons move through their own electron transport system, again pumping hydrogen ions inside the photosynthetic membrane. Eventually, the photosystem 1 electrons are taken up by empty electron-carrying NADP+ molecules waiting at the end of the photosystem 1 electron transport. Each positively charged NADP molecule picks up 2 energetic electrons from the photosystem 1 electron transport and a hydrogen ion from the stroma to form an NADPH molecule. Both the NADPH and ATP molecules formed by the two photosystems are water-soluble molecules, which dissolve in the chloroplast's stroma and remain there to be used later to supply the energy for the light-independent dark reactions. We have just seen the flow of electrons from the reaction center of photosystem 2 through photosystem 2's electron transport system to the photosystem 1 reaction center, where they replace the electrons the photosystem 1 reaction center has given up to the photosystem 1 electron transport system in order to pump hydrogen ions across the photosynthetic membrane and form NADPH molecules. In order for this flow of electrons to be sustained, a photosystem 2's reaction center must be continuously supplied with new electrons to replace the ones that it gives up. These replacement electrons come from water molecules. Photosystem 2's reaction center captures electrons from H2O molecules from within the thycaloid compartment. As a result, the H2O molecules break apart. The broken water molecules liberate hydrogen ions and oxygen atoms. The hydrogen ions increase the number of positively charged ions in the thycaloid compartment available to drive the synthesis of ATP molecules. The oxygen atoms combine to form O2 molecules, which may be retained by the plant for cellular respiration or given off to the atmosphere, where it can be used by animals for the same purpose. The reactions we just looked at are called light reactions because they require light energy to power them. The reactions that we are going to look at next, in which glucose or other molecules are actually synthesized, are referred to as dark reactions because they do not need light energy to power them. Rather, dark reactions obtain their energy from ATP and NADPH molecules formed by light reactions and then released into the stroma, where the dark reactions take place. The synthesis of glucose and the capture of the carbon dioxide molecules critical to their formation occur in a set of dark reactions known both as the Calvin-Benson cycle, after the discoverers of the cycle, and the C3 cycle, because some of the important molecules in the cycle have 3 carbon atoms in them. We'll refer to it as the C3 cycle, as that name is both shorter and more descriptive. The 3C cycle requires 4 basic components: CO2 from the atmosphere, a CO2 capturing sugar, ribulose bisphosphate or RUBP for short, enzymes to carry out and catalyze reactions, and energy in the form of ATP and NADPH molecules to power the reactions. The C3 cycle can be thought of as being divided into 3 parts: carbon fixation, PGAL and glucose synthesis, and RUBP replacement. In order to form a 6-carbon glucose molecule, 6 carbon atoms must be captured from carbon dioxide molecules. So as each C3 cycle begins, 6 5-carbon RUBP molecules combine with 6 CO2 molecules to form 6 extremely unstable 6-carbon molecules. These molecules spontaneously react with 6 H2O molecules and break apart to form 12 3-carbon molecules of phosphoglyceric acid, or PGA for short. This stage is referred to as the carbon fixation stage, as during this stage, gaseous inorganic CO2 molecules are fixed into relatively stable organic PGA molecules. In the step after carbon fixation, PGAL and glucose synthesis, energy provided by 12 ATP and 12 NADPH molecules is used in a series of enzyme-catalyzed reactions to synthesize 12 PGAL molecules from 12 PGA molecules. PGAL is a versatile molecule that can be converted into a number of different molecules, depending on the needs of the plant. Not only can 2 PGAL molecules with 3 carbons each combine to form a 6-carbon glucose molecule, the PGAL molecules may also be used to synthesize lipids, amino acids, or parts of nucleic acids. Thus, the PGAL molecules, synthesized in the C3 cycle, provide the raw materials to produce almost everything needed by a cell. In order for C3 cycles to continue on in a continuous loop, however, the 6 RUBP molecules the C3 cycle started with must be regenerated. Thus, 10 out of the 12 PGAL molecules initially synthesized are used for the regeneration of RUBP molecules, meaning that only 2 of the 12 PGAL molecules is available to synthesize glucose or other molecules. Through a complex series of reactions requiring energy from 6 ATP molecules, the 10 PGAL molecules unused in glucose synthesis are converted back into 6 RUPB molecules so that a new C3 cycle can begin. As we've seen, atmospheric carbon dioxide is critical to photosynthesis. Carbon dioxide, as you will recall, enters the leaves of plants through small pores called stomata. But when the stomata are opened up to bring in carbon dioxide, another material critical to photosynthesis, water, escapes through evaporation during hot, dry conditions. Thus, in hot, dry conditions, the C3 plants we just looked at close their stomata so water can't escape. Photosynthesis continues for a while, and one of its byproducts, oxygen, builds up in the leaf. The oxygen buildup occurs because oxygen, like water, can only exit the leaf through the stomata. Eventually, carbon dioxide levels in the leaf drop dramatically, and photosynthesis comes to a crawl as the O2 molecules, rather than carbon dioxide molecules, bind to the RUBP molecules that initiate the C3 cycle. This binding of O2 molecules to RUBP molecules leads to a series of chemical reactions called photorespiration. Unlike photosynthesis, photorespiration doesn't produce high-energy molecules like ATP or NADPH and is thus of little or no use to the plant. In order to overcome the problems created by hot, dry weather, two major adaptations have evolved that allow plants with these adaptations to have adequate levels of carbon dioxide available for photosynthesis even in hot, dry conditions. One of these adaptations is found in C4 plants, the other in CAM plants. In C4 plants, such as corn and sugarcane, carbon is first captured as 3-carbon PEP molecules in the cells of the mesophyll react with carbon dioxide molecules from the atmosphere to form the 4-carbon oxaloacetate molecules that give C4 plants their name. Unlike RUBP molecules, PEP molecules never attach to O2 molecules, no matter how high their concentration, and continue to attach to carbon dioxide molecules even when CO2 concentrations are very low. This means C4 plants don't have to open their stomata as often or as wide as C3 plants to let in carbon dioxide and expel excess oxygen. As a result, C4 plants lose less water in hot, dry weather. The advantage of C4 plants in hot, dry weather is further enhanced by the fact that photosynthetic activity in these plants is focused in the bundle sheath cells that surround the water-carrying veins of the leaf. A high concentration of carbon dioxide is ensured in the bundle sheath cells of C4 plants by a constant flow of oxaloacetate molecules from surrounding cells, which break down and release their captured CO2 once they arrive in the bundle sheath cells. The bundle sheath cells use the abundance of carbon dioxide to proceed through a regular Calvin-Benson or C3 cycle. The one drawback to capturing carbon dioxide the way C4 plants do is that once oxaloacetate molecules give up their carbon dioxide, the remnant molecule requires energy from an ATP molecule to build a new PEP molecule that can again capture carbon dioxide. Thus, in conditions where there is sufficient water, C3s outcompete C4 plants because they don't have the energy expense incurred in capturing carbon dioxide twice. C4 plants are not only outcompeted in wet and cool environments by C3 plants, they are also outcompeted in extremely hot and dry conditions by CAM plants. Saguaro and prickly pear cactus of America's desert southwest are examples of CAM plants. CAM is short for crassulacean acid metabolism. CAM plants fight water loss by only opening their stomata at night. The carbon dioxide captured at night is used to form malic acid that is stored in the central vacuole of cells until the next day, when it breaks down and releases its stored carbon dioxide. Just as in C4 plants, a regular C3 begins in a carbon dioxide-rich environment. During severe droughts, some CAM plants never open their pores and simply use the small amounts of carbon dioxide they produce internally during cellular respiration to carry out photosynthesis. Thus, CAM plants, like desert succulents and the Spanish moss that drapes many trees in the American South, can outcompete both C3 and C4 plants in a variety of extreme and unusual environments. But their method of capturing carbon dioxide causes CAM plants to be very slow growing and outcompeted by both C3 and C4 plants in more moderate environments. How plants obtain the energy and materials necessary to support their growth and reproduction had long been one of the great mysteries of science. Even though the mystery now has been largely unraveled, one can't look at the great forests and grasslands of the earth without a sense of wonder at the process of photosynthesis that turns sunlight and a few simple molecules into life. As a result of the experiments by Van Helmont, Priestley, Ingenhousz, and more recent scientists, it became obvious trees and other plants obtain the matter and energy needed to sustain growth through chemical reactions involving water from the soil and carbon dioxide from the atmosphere, and that these reactions are powered by light energy and produce oxygen as a byproduct.